Hi, this is Dr. Collins Jones. Welcome to segment 2 of my video series on manual cell counting and cell density calculations. In this segment, we will review the basic process of cell counting and get an introduction to the hemocytometer. Just to review from the previous segment, if we want to do cell counting, the first thing we have to do is get an aliquot of cells to count. In this slide, I'm illustrating a culture of suspension cells. So these cells are growing as suspension. They are not attached, and so we can simply remove an aliquot. But remember, you're going to take these off the shaker, put this into the BSC, and during that time, there may be some settling of the cells. So before you remove an aliquot, make sure that you agitate the flask to be sure the cells are uniformly dispersed, that they're uniformly suspended throughout the flask. Generally, we take a 0.5 ml aliquot of cells to count. Some people take more, some people take less. If you're using an automated cell counter, use the manufacturer's directions as to what volume of cells need to be loaded into that automatic cell counter. But again, be sure that if you're sampling a suspension cell culture, that the cells are uniformly suspended throughout the flask. And just as a reminder, if you're working with anchorage-dependent cells, the anchorage-dependent cells need to be detached before you remove a sample for cell counting. This is a common error that people new to counting and new to cell culture make, is they forget to detach the cells both to count and to passage. So if they're anchorage-dependent, you need to detach the cells, usually using trypsin, but again, as we said before, sometimes using a scraper. Okay, so we have removed our 0.5 ml sample of cells. Generally, I do this using a 2 ml sterile serological pipette, and I pipette those cells into a sterile, pyrogen-free microcentrifuge tube. So here they go. We've put them in the tube, and again, this process is done aseptically inside the BSC because the cells that are remaining in the shake flask will be passaged to new flask, and we don't want to contaminate them. So obtaining the sample for counting is done aseptically inside the BSC. Once we have our cells in our microcentrifuge tube, we can remove that tube from the BSC and do the remainder of this on the benchtop if you wish. And so the next steps would be to dilute the cells with a balanced salt solution and then to add trypan blue. Generally, I do this in additional microcentrifuge tubes. I leave the original sample intact in case my dilution is not correct, or that is, there's too many cells or too little cells to count, or something goes wrong with the trypan blue incubation. So I'd just like to have some backup, but that's just how I do the counting. Okay, so we're going to remove some cells. They're going to be diluted with balanced salt solution. Then we're going to add trypan blue. And again, I would mention that some labs, some companies do every dilution with trypan blue. We don't recommend that because trypan blue is not completely soluble. And the more trypan blue you put into your counting mixture, the more likely you are to have some undissolved trypan blue crystals, which may be accidentally counted as dead cells. So we always just do 1 volume of trypan blue with 1 volume of cells to count. That is a 1-to-2 dilution. Okay, we incubate for 2 minutes per the manufacturer's instructions. Remember, 2 minutes gives enough time for the trypan blue to enter the dead cells whose membranes are presumably compromised. That is, the membranes are leaky in the dead cells, so trypan blue can enter into the cells. The live cells, the membranes are intact, so they exclude the dye. That is, the dye cannot enter into the live cells. But again, if you incubate for too long, trypan blue can actually enter into live cells. So we recommend the 2-minute incubation with trypan blue. We then, and if there are any dead cells, then you will see some blue cells when you look under the microscope. We then load the hemocytometer and begin the counting process. Okay? And so that's what that looks like. All of this is summarized in words in the panel on the right-hand side. I'm not going to reread that to you, but I do want to emphasize that once we have added the trypan blue, the counting should be completed within 5 minutes. So you have a 2-minute incubation, and that gives you 3 minutes to count the cells. It could take a little longer, but you really want to count the cells as quickly as possible. And again, this part, the dilution, adding the trypan blue, and certainly the counting with the hemocytometer, can be done on the bench outside of the BSC. And remember that these samples are biohazard waste, and trypan blue is also considered to be chemical waste, so be sure that you dispose of these properly. All right, let's look at a hemocytometer. This is a picture of the improved Neubauer hemocytometer. And I just wanted to point out, these are not cheap, right? They range anywhere from $50 to almost $400 depending on the quality of the hemocytometer that you wish to purchase. These are made out of glass. If you drop them on the floor, they will break. Trust me, I've seen this happen. Okay, so you want to handle these with care, and you also want to be sure that you don't scratch them. So in segment 1, we talked about the counting chambers. Right? So these silvered parts on the hemocytometer, these are the counting chambers. And then if you notice, when you look under the microscope, you will see squares etched in the chamber, and we will discuss that in the next couple of slides and also in segment 3. And then if you notice here, there are some notches and on the counting chambers, and that is where you load your cell suspension. So you're going to place the tip of a P20 or P200 micropipette into this notch and then gently expel the cells into the counting chamber. Right? And this is really, really important because how you load the chamber affects the accuracy of the count, and that's where we're going to move over here to the right side of this particular slide and talk about the cover slip, which is right here. And if you recall in segment 1, I mentioned that we should not throw out the cover slips. They are not disposable. They actually cost up to $10 a piece because they are precisely made to have a specific weight. And so what happens here is that when you put the cover slip over the counting chambers before you load any cells, right? You place the cover slip on the counting chamber, and then you add the cells between the actual hemocytometer chamber and the cover slip, so the cells fit in between the two, and there should be here a clearance, if you've loaded correctly, of 0.1 millimeters. And so why am I making such a big deal of this? Because this hemocytometer is engineered that when you load the counting chamber correctly with the cover slip already on, right? So you're squeezing the cells in between the counting chamber and the cover slip. This then corresponds to a specific volume, length times width times depth, or depth is height. So length times width times height, right, is a cubic measurement, and that is a volume. So if you do not load the hemocytometer correctly, you don't have the correct volume and your counts are not accurate. Okay, this is what the squares, the etchings on the hemocytometer should look like under the microscope. So it's really important that you learn to get a clear view of these, that you focus correctly on the hemocytometer to do the counting. And it's worthwhile, if you're just learning to count or if you haven't counted manually for a while, to just take an empty hemocytometer and practice focusing on these grids. Okay. Now we talked about the counting chamber. This is really 2 counting chambers, the entire chamber. But then we talked about squares within the chamber. So a single square is this, right? And the lines are etched outside, and that's just part of the process of the manufacturer. But this is a single square. But you may notice, I hope you notice, that within that so-called single square, there are 16 smaller squares. So the large single square is made up of 16 smaller squares. And this is done just to make it easy to count the cells, so you don't count the same cell twice, so you can keep track of where you're counting, and we'll illustrate how to do that in the next segment. Okay, so we have this single square composed of smaller squares for the ease of counting, but we also mentioned that you should count 10 of these squares total, that is 5 per side. So here's what the 5 large squares for counting look like per side. Okay, And so we go, and we'll again illustrate this in the next segment. 1, 2, 3, 4, 5. And you may notice this is a little more difficult to view because originally these hemocytometers were designed to count blood cells, and you have many different kinds of blood cells: red blood cells, white blood cells, and then there are different types of white blood cells, and they're all different sizes. And so the hemocytometer was designed to be able to count all those different types of blood cells, which are of different sizes. If you're doing cell culture, then all of your cells should be exactly the same. That is, if you're growing CHO cells, all the cells you're counting should be CHO cells. If you're growing 3T3 cells, they should all be 3T3. If you're growing HEK293s, they should all be HEK293s. SF9s, which is one of the cells that we grow in our lab, those are insect cells, they're all going to be SF9 cells. All right, and so this really is not necessary, but it's just the way they sell the hemocytometer to get a bigger market for both people who need to count blood cells and people who are going to count cells in cell culture. All right, so loading the hemocytometer. Remember I said this is really important, right? So we're going to obtain the hemocytometer and the cover slip. Make sure the hemocytometer and the cover slips are clean and dry. There's no residue from it. And really what I tend to do is inspect the hemocytometer under the microscope before I load it, before I use it, to make sure that the last user cleaned it properly and there's no debris or particulate material stuck on the hemocytometer. Can't emphasize this enough. Please do not throw out the cover slips, right? And so here they are over here. Make sure they are clean as well. Before you load the cells, you're going to place the cover slip over the counting chambers, that is over the grid. And then you're going to take your cells that were in the microcentrifuge tube that have been incubating with the trypan blue for not more than 2 minutes. And you're gently going to mix these by trituration, that is just drawing up and down very slowly in the micropipetter. We want to avoid shear, but we want to make sure that we have a uniform suspension of the cells. Remember, the cells settle over time. And then we recommend that you draw up about 20 to 30 microliters of suspended cells. I always use a P200. This is more volume than I need, but it's just easier to work with, okay? Make sure that there are no bubbles when you draw up the cells. Bubbles are troubles, right? Then carefully pipette the cell suspension between the cover slip and the counting grid, okay? So again, the tip is placed in the notch right here, and then we're very slowly and steadily, we're going to expel the cell suspension into the chamber, and we're going to be careful not to overfill or to underfill. All right. And so this again takes a little bit of practice, right? And going slowly and steadily is part of the key to being able to accurately fill the hemocytometer. Okay. So if we look here, let's look at hemocytometer loading errors. If we've loaded the hemocytometer correctly, the counting chamber will be full, but it won't be overflowing. All right. And so I tried to take a picture here with a real hemocytometer, but it didn't come out too well. But you can see, hopefully you can see what a correctly filled hemocytometer looks like with real trypan blue and cells in suspension. So one error that people make when filling the hemocytometer is they underfill it. So we haven't put enough of the cell suspension in the trypan blue in the hemocytometer. It's only partially filled, all right? And this doesn't work. We don't have enough volume of cells in there. We don't get the right volume. This will be an inaccurate count. And again, here's what a partially filled hemocytometer may look like, okay? And then conversely, if we go a little bit overboard and we push too fast or we try to load all 30 microliters, then we're going to overfill the hemocytometer and the cells that we are loading are going to spill out onto either side and maybe even behind. And again, the cover slip will not sit correctly on the counting chamber and the volume will be incorrect and the counts will be inaccurate. Okay? All right. So one thing that we use to kind of makes it easier is something called the cellometer, right? This is a commercially available product. It's sold by Nexlom. It sounds like I'm endorsing it. I like it. And there are probably other versions on the market, but this is the one that we've been using for years. So the two things I like about this is the cover slip and the counting chamber are already sealed together at the right depth, right? So we don't have to worry about the placement of the cover slip, and to a certain extent, we don't have to worry a lot about overfilling or underfilling. The other thing I like is that this is a disposable counting chamber, so that if we're using anything that's pathogenic or potentially dangerous, we can just throw out the entire salometer counting chamber into the biohazard bag and not worry about it. There's no cleaning when you're done. You just discard this. This is a single-use hemocytometer. So the advantages are it's easy to load. If you overload this, it just comes out the other side, right? Which isn't preferable, but you can kind of get away. I've tried this. You can get away with slightly overloading it and still get accurate counts if you compare the slightly overloaded to correctly loaded. The grids are really easy to see, and again, this is single use, so you don't need to worry about cleaning. You don't need to worry about cross-contamination. If you're working with pathogenic substances, it's easy to discard. The disadvantages are, even after 10 years, not everyone uses this, so it's not universally accepted for use. You probably need to do some validation with our Neubauer hemocytometer versus the Celometer. And I have found that the slides really should be stored in the box they're shipped in, of course, but that it comes with a desiccant, and you may need to reactivate that desiccant. Otherwise, if there's a lot of humidity, it impedes the proper loading of the Celometer. Okay, and so again, loading the Celometer is very similar to loading the hemocytometer. You obtain the cell suspension mixed to ensure a uniform suspension. Again, draw up about 30 microliters, and then slowly and steadily pipette this into the sample introduction port. It's really easy to see, and you can see this just spread out really nicely through the chamber, right? And so here again, I've taken some pictures. You can see this a little bit more easily. This is a Celometer that is correctly loaded. This is underfilled, and so this will not give you an accurate count. This is overfilled, and you can see that there's little bubbles of trypan blue coming out the top. This is not ideal because you may get dye, especially if you're using a semi-automated counter like a Countess or a TC20. You may actually end up getting dye on the lens in that unit. So be careful. They sell sort of automated versions of this. So be careful not to overload it. And again, just to look at the setup here, once the hemocytometer is loaded, we're going to place it on the inverted microscope. We're then going to focus this so that the grid lines and the cells can easily be seen. There's a sharpness to this, right? And then we have a manual cell counter right next to the hemocytometer, or right next to the inverted microscope. These are ancient but still very functional. And then we're going to focus on the cells and be able to see this large square composed of 16 smaller squares. And we're going to count live cells, which should be translucent or clear. You should just be able to see the outline of the membrane. And simultaneously, we're going to count dead cells, which will be blue. And so that's why we have the two buttons here. So generally for me, I put my fingers over this, right, index finger and forefinger. And so the red button is for live cells. So every time I see a red button, I push this down and get a click. And the white button is for dead cells. And then over here on the side, if this unit is working correctly, this is the total number of cells. So it adds this plus this to give you the total number of cells. The one thing when you use this manual cell counter is to check to make sure that you have set it to 0, and you set this to 0 by rotating these red knobs on either side, but make sure all of the windows read 000 before you start your count. And that is the end of segment 2.